Hey guys, welcome to Man Medicine, where we talk about how men can optimize their health and escape the collapsing U.S. healthcare system. Well, today I've been meaning to give you guys another deep dive for quite some time, and I just have not been able to get around to it. So today I'm going to make up for that. We're going to do part two, or I guess the next in the series that I'm going to call deep dives. And today we're going to do it on the androgen receptor. The androgen receptor, this is an incredibly complicated topic, so I'm probably going to need to do multiple parts to this. Otherwise, I'm going to be here for hours. I mean, there are people that do their entire PhDs on the androgen receptor. So I want to focus in on what I think are some really important clinically relevant details about the androgen receptor that you can take home if you're a clinician. Now, as a reminder, like these deep dives, I understand these are not for everybody, okay? In these deep dive sessions, I'm going to be presenting you guys with very in-depth, very detailed information on a specific subject related to men's health. And it's going to be presented at essentially the medical school level and above, okay? So yeah, I'm going to assume that the people that are going to watch this video have already got a certain background, at least in, let's just say college level biology, anatomy, physiology, whatever the case may be. Things are going to be a lot more specific and a lot more detailed in this than in my general content. And I understand that's not for everybody. And if it's not for you, that's totally cool. Like, you don't have to watch this. But I like to do this every now and then because I just don't see very much of it on YouTube. And this kind of high-level material in the world of TRT, I think, is sorely lacking. Nobody either knows about it or talks about it outside of just very specific circles in endocrinology and andrology, and it's important. I think if you're going to prescribe testosterone therapeutically to men and to women for that matter, to hormones in general, that you really need to have this doctorate level understanding of how these medications work. I mean, I was always taught in medical school, and it was drilled into me in residency that, you know, you're not allowed to prescribe a medication until you thoroughly understand its mechanism, its indications, its contraindications, its side effects, and you know, certainly that would apply to testosterone. It applies to thyroid hormone. It applies to anything. So we're going to take a deep dive on the androgen receptor today because I think it's really important and it's actually pretty cool. So, all right. So why does learning this stuff actually matter? Well, this actually has really important clinical implications. One of the things that's a bit frustrating in the world of, I'm sure any endocrinologist will tell you this, it's frustrating to me. I'm not an endocrinologist, but it's frustrating to me when it comes to, you know, treating men's hormones, is really we just have two things to go on when we're monitoring patients, and one of them is serum blood levels of whatever it is we're looking for. Estradiol levels, testosterone levels, thyroid, you know, T4, T3, and then we have the clinical response of the patient. But everything in the middle there, even though in many cases we understand the mechanism in great detail, there's really no practical way to monitor or evaluate that process, and specifically, there's no way to identify individual differences that the patient who's actually sitting in front of you might have that someone else doesn't have. And I think understanding that's really important, because what you'll find is, as you go through your practice over the years, is that you're going to see people that either are like non-responders in some cases, or they're hyper responders, and they may be on identical protocols. And the reason for that is genetic differences, and it's somewhere in that middle black box space. Because you know, I've seen it many times. I'm sure any of you who've prescribed testosterone have. We'll just use testosterone as an example. You can have two men who outwardly appear very similar age, similar demographics, and you can give them identical doses of testosterone. You can even achieve very near identical serum levels of testosterone, and you can have widely variable clinical effects. You may have one who feels absolutely fabulous, and all of the parameters that caused you to prescribed testosterone in the first place have all gotten better, and then the other guy just is sort of has a lackluster response, or has no response, or even has a negative response. Well, why is that? Well, it's complicated, but a big part of it, not the only part of it, but a big part of it is likely related to the androgen receptor when it comes to testosterone. So that's why I want to talk about it. It's important to understand that there are wide genetic variations in the androgen receptor, as I'm going to show you, and it does have clinical implications. Okay, let's talk real quick about just how the androgen receptor works, how it gets activated, just the basics. Okay, so the androgen receptor, it floats around in the cytosol, which is inside the cell. It's not really found free floating in the serum to any significant extent. I mean, it is a little bit, but it's mainly intracellular, and it sits there, and it has something called heat shock protein number 90 attached to it. Okay, and what that heat shock protein does is it keeps the androgen receptor in a conformation that is open to bind to its ligand, which, you know, for all practical purposes, the vast majority of the time is going to be testosterone or DHT. Although, you know, there's other weak androgens that can also bind to the androgen receptor. Androstenedione, DHEA can bind very weakly. There's a whole bunch of other ones, but, you know, we're just, for the sake of this talk, we'll just assume it's either going to be DHT or testosterone. DHT has a higher binding affinity, mainly just because it's easier for testosterone to dissociate. So that, you know, it binds both of them quite well, but testosterone is able to escape and to release itself from the androgen receptor a little bit more easily than DHT does. So anyway, DHT or testosterone comes along, it binds to the androgen receptor, it induces a conformational change in the protein itself. So this is a picture of the protein here, a really cool, like, multicolored 3D one that I saw on a site called Fine Art America, which is actually a really cool site. They sell some really nice fine art, so you should go check them out. I am not sponsored by them. But I like this one so much, I might actually buy this for my office. But anyway, so this heat shock protein number 90 is ejected, essentially. It's released. And so that allows two proteins, important alpha and important beta, okay, to attach onto the androgen receptor complex. And then that's what allows it to be transported through these pores in the nucleus, these channels, which are tightly regulated. So that complex then, it floats through those pores into the nucleus, okay, that's where things start to get interesting, okay? So once it's in there, for reasons I'm not quite sure why, but this is just the way that it's designed or evolved, the androgen receptor, it links up with another androgen receptor. So now you have this dimer, okay? So the two of those, now these are ready to then bind onto DNA and to initiate transcription and do all the things that androgens do, okay? The interesting thing is, though, that there's a host of these, the reason I said it gets interesting and complicated is there are a host of these co-regulators that some of them assist and encourage or stimulate the binding of this dimer to the various genes that it's supposed to bind to, and then others actually inhibit those. And so you have this interplay with all these different co-regulators that, even though the androgen receptor complex is bound to DNA, depending on what these co-regulators are doing and in what concentrations they are, you know, you can have a lot of DNA transcription, or you might have very little at all. And these co-regulators, just to make things even more complicated, are influenced strongly by epigenetics, which we're not going to get into today. But you can see how you can really go down the rabbit hole with this stuff. And the thing I want you to remember through this entire talk is all of these different proteins, all of these, you know, these co-regulators, this androgen receptor, the nuclear pores, you know, the important alpha, important beta. You know, all of these are also encoded by genes. And again, all of which may or may not have individual genetic variations from person to person, which affect the way that they work. So all of this together is incredibly complex. You have to take this into account when you are trying to assess somebody's clinical response to to testosterone or to anything else for that matter. So things start to get very, very complicated, and this is why I really do not like algorithmic cookie cutter approaches to anything in medicine, but you know, particularly we're talking about TRT here, and why I get a little frustrated with, for example, like the Endocrine Society guidelines when it comes to you know, the treatment of hypogonadism, because there are so many things that go into, you know, that go into the symptoms of hypogonadism. There's so many things that play into the effectiveness of testosterone therapy and the symptoms that a man might have that go well beyond what their serum levels are. You know, serum levels are not irrelevant, but oh my gosh, there's so many other things that are important that, you know, as I'm telling you here, and what we'll see, it's going to get even more complicated here in a second, that, you know, basing a decision to treat purely on a serum level, which really is done commonly. Now, the Endocrine Society doesn't advocate that. They want clinical symptoms as well, obviously, so I don't want to, you know, say the wrong thing in that regard. But, you know, there are clinics out there that just look at your number and be like, hey, man, you're low. You need to be on testosterone, and that's not necessarily the case. Okay, so that's basically how the androgen receptor works. It binds to your DNA, and it causes DNA transcription. Now, maybe this might be for another deep dive later. There is a whole other non-DNA binding mechanism that the androgen receptor also activates. And it does so much more quickly through a second messenger system. So, you know, anything that, any steroid that induces DNA synthesis, you know, it's a slow process, right? It may take several hours, but second messenger systems are designed to be very quickly, sometimes just within a few seconds, maybe a few minutes. Well, it turns out there is a pathway for this androgen receptor to make things happen much more quickly through this non-DNA binding mechanism that I'm not going to get into today. Again, maybe I'll do it on another deep dive, but it has these two different broad pathways in order that it uses to exert its clinical effects. Okay, this is what I'm trying to say. So we're not going to worry about the second one today, because otherwise this would be way too long. Okay, let's talk about the genetics of the androgen receptor, just briefly here. So the androgen receptor, the genes for the androgen receptor are located on the X chromosome. So as a male, you have an X chromosome and you have a Y chromosome. Women have two X chromosomes. Okay, now, so that means you only have one copy. So that could be a problem if you have a defective androgen receptor, because you don't have a backup that works. And so we'll talk about what happens when you have a defective androgen receptor. But the point is that you inherit your androgen receptor from your mother. It does not come from your father. It's not on the Y chromosome. Okay, so the gene for the androgen receptor, so it has 8 different exons, all of which have introns in the middle. Okay, but these 8 different exons, when they are translated, they come together and they make the androgen receptor protein. Okay, so the transactivation domain, which we're going to talk about a lot, is encoded by exon 1, so that's over half of it. Exon 2 and 3 encode the DNA binding domain, which we're not going to talk about as much. A portion of exon 4, the 5' portion, is what encodes the hinge region, which is a tiny little region that's actually really, really important. We'll talk briefly about that. And then the rest of the exons, like 4 through 8, they encode the ligand binding domain. So that's the little, the ligand binding domain. It's this little hydrophobic pocket where androgen binds. So it's where DHT and testosterone bind. It's about 24 amino acids. It's a little pocket, and that's where the ligand goes in, and then the conformational changes that I alluded to earlier begin to happen. So the protein itself, so the androgen receptor protein, it's 919 amino acids. Some fun trivia for you. And it has 3 major functional domains that I just mentioned. We'll talk some more about those in a second. Now, the interesting thing is, so the androgen receptor is, the structure of the androgen receptor is very similar to other steroid receptors in the body. So it's highly conserved throughout the body. And interestingly enough, it's highly conserved in all mammals. So there are obviously differences between, you know, humans and hamsters. But if you look at the way that their steroid receptors are structured, they look very similar. You can tell that at some point there was a common ancestor there. Okay, so the closest thing to the androgen receptor is actually the progesterone receptor, which is actually like 88% identical. So it looks very, very similar, but glucocorticoid receptor, the mineralocorticoid receptors throughout the body all look very similar. They're about 50% identical in terms of their DNA sequence to the androgen receptor. So they're all kind of variations on a theme, and you know, they're selective for different steroids, but they all kind of work in roughly the same way. Real briefly, I want to talk about some of the more like really serious mutations in the androgen receptor, of which there are many, but we're mainly going to talk about like very subtle androgen receptor mutations that you know, that have clinical relevance for men on TRT and men in terms of their overall health. But you know, there are some more serious ones that like completely obliterate the ability of the androgen receptor to bind onto androgen or limit it like extremely. And so those individuals end up with something called complete androgen insensitivity. We used to call it testicular feminization syndrome. And those people generally end up what we would, you know, typically call intersexed. So as they grow up, phenotypically, they're XY in terms of their genetics, but phenotypically they grow up looking female. The only difference is they tend to they don't have a uterus. They have, you know, kind of a poorly formed, they don't have a fully formed vagina, and so their internal organs are not fully developed, and they don't go through puberty normally. But externally, and certainly once they're put on hormones, their phenotype is female, and that's because their androgen receptor it cannot respond to testosterone. So in utero, as a fetus, you know, the default pathway in embryology for the human embryo is female, and only in the presence of testosterone and DHT does an embryo begin to take on its male characteristics. So if you got a receptor that cannot respond to androgen, it just follows the default pathway, which is female. Okay, we're not going to talk about that so much, but that's just so you understand. Okay, let's get into the structure of the androgen receptor a little bit. So as I mentioned, there's the three functional domains that are really, really important, and there's this N-terminal domain, and we're going to spend most of our time talking about the genetic variations there, because this is where you get the most variation among individuals. So, and then you have the DNA binding domain here in green, the hinge section, and then the ligand binding domain. Now there are genetic differences in these areas, but these areas tend to be a little bit more conserved evolutionarily, because when you have problems in those areas, you have major problems, and natural selection is going to weed you out of the population if you have major mutations in those areas. The N-terminal domain, you've got a little bit of leeway there. Not a lot, but you do have some. Okay, so the hinge area, let me touch on that briefly. The hinge area is really complicated. It has a lot of different actions. It's mainly involved in DNA binding and nuclear translocation signaling, and about 4 or 5 other things that I just don't want to get into right now. But it is really, really important. If you have major mutations in the hinge area, you've got big problems. I already mentioned the ligand binding domain. That's that little hydrophobic pocket in there that actually like holds on to the testosterone and to the DHT. But let's talk about this N-terminal domain. And there's 3 things that happen in this area that I want to talk about that have direct clinical relevance to testosterone therapy in men. And many physicians that prescribe testosterone are aware that there are these called CAG Repeats, CAG, which is cytosine, adenine, and guanine, those 3 nucleotides, they encode glutamine. So you have these long like polyglutamine chains essentially at the N terminus, and depending on how many of those you have, it will influence how well androgens will bind to the testosterone. It has, it does have significant clinical clinical relevance. So, if you look at population studies, most men have somewhere between 9 and 36 of these polyglutamine repeats. Okay, there are racial and ethnic differences. I'll talk about that in just a second. But if you just, sample everybody, men around the world, this is what you're going to find is a vast majority are somewhere between like 9 and 36, usually clustered around in the low 20s. Okay, but what a lot of docs don't know is that there are also there's two other areas where there are nucleotide repeats that are just as important as these CAG repeats. It's just that they're not as well studied. We don't really understand them as well. The CAG repeats, people start to figure those out at this point. And there are even some tests that are very expensive where you can find out how many CAG repeats you have. But as far as I know, there are no commercially available tests for these other ones. So the first one is a TGG repeat. Okay, so that's thymine, guanine, guanine. That encodes proline. Okay, which is another amino acid. So you have these polyproline chains also at the N-terminal domain of your androgen receptor. The average there is about 8, okay, for most men. And then this other one is also, it probably is of equal importance to the CAG repeats. It's this GGC, guanine, guanine, and cytosine, which codes for glycine. So you have these polyglycine chains. And there's a bit of variability here, just like with the CAG repeats, somewhere between 10 and 27 of these polyglycine chains. And this does have significant influence on androgen binding as well, just like the CAG proteins do. So that's the structure of the androgen receptor. We're going to talk a little bit more about the CAG stuff here because that's important. We'll touch on the other ones as well. Just briefly, I do want to mention the DNA binding domain. This is obviously really important. This is where the rubber meets the road, so to speak. So this looks very similar to all the other steroid receptors that are in your body, as I mentioned. And they have the ubiquitous, if you remember from your chemistry, the zinc fingers. So zinc fingers are involved in DNA binding. They're highly conserved throughout all species on Earth that have steroid receptors, so actually crucially important. So the DNA binding domain on the androgen receptor, it's like 70 amino acids long, and so that you can see it consists actually of, there's 2 zinc fingers in there, and I'm not going to go into zinc fingers, but this is a structure of a zinc finger. You have a zinc atom that's held in place here by these, on one side there's 4 cysteines, and then another 4 cysteines on the other side. You have your zinc finger in the middle, and then there's actually another one that's like right next to it. Okay, so that's the DNA binding domain. Let's get back to these CAG repeats, because this is actually kind of interesting. So these CAG repeats are encoded by exon number 1, as I mentioned earlier, and if the more of these you have, the less effective your androgen receptor is in terms of binding testosterone. And as I mentioned, there's a lot of variability in the normal amount, but the average is somewhere between 22 and 23. So like I said, low 20s, although again, 8 to 36, 38, something like that, if you look at men all over the world. What's interesting though is that there's a certain type of muscular dystrophy called Kennedy's disease, where you have a lot of these CAG repeats, like many, like over 60 in some cases. And so that obviously presents a real problem, and over time it's kind of a slow and progressive disease. The other name for it is spinal bulbar muscular atrophy. I learned it as Kennedy's disease. I think it still gets called that by old timers like me. But it is, it's this progressive degenerative neuromuscular disorder. You get wasting of your proximal muscles, but also your bulbar muscles. So you'll see when they stick out their tongue, there's like atrophy of their tongue. And not surprisingly, because they've got these long CAG repeats, they also suffer from signs and symptoms of profound hypogonadism. So it's very common for them to get testicular atrophy, to develop gynecomastia, as you can see in the picture here. You can see the tongue as well, like pretty severe gynecomastia. They have low fertility, or they're completely infertile in some cases, and that's a result of these long CAG repeats. Unfortunately, they develop severe, as time goes on, they develop a lot of severe muscle symptoms. Many of them end up wheelchair bound, so it's pretty sad. The other interesting thing about these CAG repeats, I remember when I learned about this in regards to the androgen receptor, it triggered something that I learned about. I was like, isn't that what happens with Huntington's disease? And it turns out that's exactly what happens with Huntington's disease as well. So you have these trinucleotide repeats in Huntington's disease, these CAG repeats, many of them, which codes for this like defective Huntington's protein, which, I'm not going to go into Huntington's disease, but causes like, brain cell death, essentially neuronal death, and all of the things that happen with Huntington's disease. It's a tragic autosomal dominant disease, but it is also associated with CAG repeats. It's not involved with the androgen receptor like Kennedy's disease, but it's another disease where these CAG repeats start to pose a real problem when you get too many of them. So it's not just Kennedy's disease, it's in Huntington's as well. And there may be others that I'm not aware of, but those are the two big ones that I remember learning about. Okay, let's talk about some of the ethnic differences. This is a paper here, volume 12 of Genomics from 1992, genetic variation at 5 trimeric and tetrameric tandem repeat loci in 4 human population groups. Whew, that's a lot. Okay, so, how much clinical relevance this has, I guess is up for debate. What this study showed was that there was some slight variations if you looked at folks from folks who had African descent, European/Caucasian descent versus East Asians. And what they found was that on average, men with African descent had roughly between 18 to 20 of these CAG repeats. Caucasians had just a few more, somewhere between like 21 and 22. And then if you were of East Asian descent, the average was just a little bit higher, 22 to 23. So still like clustered very closely, but just there's some subtle differences where men of African descent tended to have just a few less on average CAG repeats than, say, folks who, let's say, were ethnically Japanese or, Han Chinese, Vietnamese, that sort of thing. So again, whether this has any clinical relevance, it's hard to say. But it's, I thought it was interesting, so I included it in the talk. So, okay, this is this is a little clip from one of the books I wrote. I took this from the World Journal of Men's Health, the 2018 edition, and basically this is just a bell curve, looking at CAG repeats. If you took a group of 1,000 men and you sequenced their DNA and you looked at their CAG repeats, this is the kind of bell curve that you would get. In the middle here, so you look at the large percentage of men, the average here, the mean, is just above 20, which is, consistent with what I was telling you guys before. But, there are outliers, right? There are always people on the left-hand side of the bell curve, and there are always people on the right side of the bell curve, no matter what characteristic you're looking at, okay? So the more you go to the right in this chart, you have decreasing sensitivity of your androgen receptor, okay? And the more you go to the left, the less CAG repeats you have, the more sensitive your receptor is to androgens. So that does have clinical implications, because if you have a very sensitive receptor, and we'll talk about this more, you will have a better androgen response for a given amount of testosterone than somebody who's on the right side of the bell curve, who has a relatively more insensitive receptor. Okay, and we'll talk about how you can pick that up. One of the things that can clue you in, it's very difficult to get these tests and sequence somebody's CAG repeats, but if it's a healthy man and they have a very high LH level, that could be a sign that they have a relatively insensitive receptor. They may have just a few more CAG repeats than average, okay? And that's because, what happens if you have an insensitive receptor, you have to overcome that by having a higher serum level of testosterone. And so how do you get a higher serum level of testosterone? Your hypothalamus dials up the HPTA axis, you secrete a higher level of LH, and as a result, ideally you'll have a higher amount of testosterone. Now, if you have testicular failure, or you have any problem anywhere in that HPT axis, you're not going to be able to do that. So if I'm referring in this case just to healthy men. And the other thing that may clue you in as well, in addition to that, is you might have slightly higher estradiol levels. Again, there's a whole genetic component to that as well, and not to mention body fat percentage. But it makes sense. Higher serum testosterone levels, you're going to have an appropriately elevated estradiol level due to aromatization. Okay, this is a really great chart. It's from this this excellent book here, Testosterone Action Deficiency and Substitution, the 4th edition. I highly recommend you check it out. This will explain to you what I was alluding to earlier. So on the x-axis, we have testosterone levels. So if you have a very insensitive receptor, meaning you have long CAG repeats, let's move to the right on that x-axis. Initially, when you're in the hypogonadal range, I mean, when you're low, you're low. And it doesn't matter if you've got a good receptor with short CAG repeats or, you know, a less, I'm not going to say a bad receptor, but a less sensitive receptor with long CAG repeats, or whether you have short CAG repeats. It's kind of irrelevant in terms of the androgen effect. But as you start going to the right and you start having higher and higher testosterone levels, if you have short CAG repeats, again, which means a sensitive receptor, you will have a higher androgen effect for a given level of testosterone. Whereas if you have an insensitive receptor, you will require higher levels of serum testosterone to get the same effect that somebody with a sensitive receptor would have, perhaps at a lower level. So it's a very simple but elegant diagram here, and I think it explains it pretty well. So what they really don't show, though, is that if you just keep going to the right, you know, eventually those curves like meet up again, because I mean you've just like completely saturated the androgen receptors. It doesn't matter how many CAG repeats you have per se. But they stopped it they stopped it in the eugonadal range. When you start going super physiologic, those two curves will meet up again, but they didn't show that. Okay, so what are the clinical implications of this? I mentioned obviously what it was in terms of testosterone, but you know, having shorter CAG repeats also has some other effects, or at least it's associated with other effects and other conditions. Whether it's causal, you know, you can argue about that, but there certainly is an association with larger prostate volumes, especially once you put a man on DHT. If you have a high number of CAG repeats, it's actually associated with lower bone density, which is kind of interesting. Also associated with higher risk of metabolic syndrome, type 2 diabetes, higher body fat percentage, and it's debatable about what the effects are in terms of cardiovascular disease. There's a lot that goes into the development of cardiovascular disease, so you know, they don't really know for sure whether, you know, the type of androgen receptor, whether that's a major contributor to cardiovascular disease. I'm sure it probably plays a small role, but it's because cardiovascular disease is so complicated, it's a little bit hard to say for now. But definitely metabolic syndrome, higher body fat levels, higher insulin levels as a result, and type 2 diabetes, definitely they are associated with longer CAG repeats. The other interesting thing, and I alluded to this earlier, is you know, low CAG repeats, but also low numbers of those GGC repeats. Those are associated with they're associated with development of prostate cancer. And the shorter your CAG repeats and the shorter your GGC repeats, there is an association with developing prostate cancer at an early age, developing a higher grade or more aggressive form of prostate cancer, and to be at a more advanced stage when you're finally diagnosed. So, it's kind of interesting. So, you know, there are clinical implications to these genetic differences that go just beyond, obviously, beyond TRT. Okay, let's switch gears and we'll talk about the TGG, which is the thymine, guanine, guanine repeats. Again, that codes for these long polyproline repeats. This is a little bit fuzzy in terms of what they do, but we do know that they're crucially important. So whenever you see these long polyproline repeats, a lot of times they're associated with proteins that have something called an SH3 domain. Now, an SH3 domain is it's a type of domain. It's found in many, many different proteins. It's conserved evolutionarily, and it's involved in communication from one protein to another specifically proteins that exist in the intracellular space, so not so much extracellularly, but very complicated. They're involved in signaling proteins to create polyprotein complexes. So they'll create these like big 3D polyprotein structures. All of those are involved with this SH3 domain, and it's thought that this TGG polyprotein repeat area has something to do with that. We don't really know if subtle mutations in this can affect androgen binding. I suspect that they probably can, because there are mutations that have been identified that result in ambiguous genitalia and like sexual underdevelopment, as I alluded to earlier. Some of those androgen insensitivity syndromes have been traced to mutations in this particular area. And there's one in particular that I found that actually reduced the effectiveness of the androgen receptor by about 80%. So yeah, it's probably pretty important. It's just we don't understand it as well as the CAG repeats. It probably does have some important clinical ramifications. Okay, we'll finish up here soon here with these GGC repeats. Again, that's guanine, and cytosine. So you get these long polyglycine repeats, average male somewhere between 10 and 27 of these. Okay, if you have longer ones of the longer repeats, so longer repeats are associated with higher risk of prostate cancer, just like the issue with CAG, but in reverse. In women, it's associated with endometrial cancer if you have these long GGC repeats. So it's kind of interesting. It affects both men and women. It definitely affects testosterone and DHT binding. So again, longer GGC repeats, less androgen affinity, you have a less effective receptor. So again, a lot of docs know about the CAG repeat influencing androgen receptor sensitivity, but many of them, including myself up until not that long ago, were not really aware that there were these other repeats, specifically GGC, that also have a pretty profound effect on androgen binding. There's one study in particular that I found, I couldn't find it again to put it up here, but it showed that if you had more than 27 of these GGC repeats, then you could reduce the activity of the androgen receptor somewhere between, they said, 37 to 78%. So it's like pretty significant. Other implications to this, again, long repeats are associated with lower sperm counts, worsening like sperm morphology and motility. Okay, that's all I want to talk about today. I want to finish up with a quote, because I really want to drive this point home. And this quote is from that book, Testosterone Action Deficiency and Substitution. I'm just going to read it because they say it better than I ever could. It says here, okay, quote, "In case of longer CAG repeats," and again, this is also going to apply to the GGC repeats as well. So, in case of lower CAG, these men will present with inadequately low normal LH concentration and or low normal testosterone levels, but nevertheless will require higher concentrations of testosterone to compensate for their attenuated androgen action. Okay, they're going to need higher doses of testosterone to get the same effect as somebody who had a more sensitive receptor. Okay, back to the quote. Thus, they are likely to present with features of hypogonadism in the presence of normal testosterone levels. Let me read that again. Okay, they are likely to present with features of hypogonadism in the presence of normal testosterone levels. Exactly this is the patient group most likely missed for investigation, diagnostics, and putative treatment up to this day. Okay, there you have it. So this is why I have a little bit of beef with the Endocrine Society's guidelines on hypogonadism. And I don't disagree with everything they say. Hypogonadism needs to be treated. But there's the implication or the insinuation there that if you don't meet the serum levels that they talk about in there, which are quite low, that there's really no indication to treat a man with hypogonadal symptoms. There's basically no possibility that treating them would make them any better. And you know, the data that I just presented to you like directly contradicts that. You know, and the fact of the matter is, you know, endocrinologists know this. You know, they know the androgen receptor backwards and forwards. It's part of their residency training. So I know that they understand that there are men who are going to have insensitive receptors, and therefore are going to have testosterone levels that are within, you know, the reference range. Yet they're going to be suffering from hypogonadism, or they're going to be suffering from symptoms of hypogonadism. So that's why, like I said, it's just really important to, you know, the old adage about treating the patient and not treating the number is just as true today as it was, you know, 100 years ago. And you know, you don't want to just dismiss somebody who's coming to you symptomatic based on a simple blood level. You want to, you know, again, you're a physician, right? So you want to do a proper history. You want to take a proper history. You want to perform the proper physical exam. You want to explore, you know, not just testosterone as the culprit, you know, because there's a lot of things that can cause hypogonadal symptoms that have nothing to do with actual hypogonadism. But you want to get to the root cause of the problem. And, you know, if you can't find anything else, and you've got somebody with a low normal testosterone level and all the symptoms of hypogonadism, well, as I just presented to you, I mean, there's overwhelming data that would suggest that it's entirely possible that this particular individual has one or more of these androgen receptor mutations that have led them to have an insensitive receptor. And, you know, that they may become symptomatic at a total testosterone of 500, whereas somebody who has a very sensitive receptor, who has very few of those CAG repeats, for example, or very few of those GGC repeats, you know, they may walk around with a total testosterone of 250 and feel absolutely fine. And I've seen those guys. Those guys are out there. Any of you physicians out there that have been taking care of men for any length of time and prescribed therapeutic testosterone, I know you've seen them as well. There are men out there with shockingly low testosterone levels who are asymptomatic. Okay, the only explanation I have for that, based on this data, is that you know, they won the androgen receptor lottery, maybe, because I don't have another explanation for it. But, you know, the point is, we're all individuals. There is genetic variation between men, and so you have to take that into account. You know, this is the art of medicine. Until we have better diagnostics that can give us more information, I would love to have a test that tells me exactly what your serum testosterone level should be for optimal overall health, you know, and that'd be fabulous, because then that'd be easy. I just get you there and off you go, or, you know, help you do the lifestyle things that you need to do to get you there, and off you go. But we don't have that test, and we probably never will. So, you know, we're kind of like groping around in the dark here a little bit, in terms of treating men. But I just think it's important to not have blinders on, not focus excessively on serum levels. Serum levels are important. I'm not going to downplay that, but there's a lot more that goes in to a man's clinical picture than just his serum levels on a given day. So that's all I have for you guys on this deep dive. I hope you liked it. Again, even though this is kind of a deep dive, barely scratching the surface on the androgen receptor, it's incredibly complex. So time allowing, maybe I'll do a part 2 on this, and it's probably going to need like a part 3 and a part 4, because it's complicated. All right. Love to hear some feedback from you guys, especially clinicians out there. Let me know what you think about this. Let me know if you have any particular clinical insights from your practice. I always love to hear those, and I will catch you guys next time. All Man Medicine video and audio has been created and shared online for informational purposes only. This podcast does not constitute the practice of medicine or professional healthcare services of any kind, including the giving of medical advice. I am not your doctor. No doctor-patient relationship has been established. This content is not meant to be a substitute for professional medical advice and should not be relied upon solely for that purpose either. The only purpose of this content is to present peer-reviewed, research-backed health information for your consideration. As always, rely on the advice and guidance of your personal physician before undertaking any activity presented here, and if in doubt or not comfortable with said activity, practice discretion. Your health is your responsibility and not ours. Finally, I take conflicts of interest seriously. I accept no compensation whatsoever from any private corporations, including pharmaceutical or supplement companies. You can trust that if I recommend a medication, product, or service, it's because I genuinely believe in it and not because I'm being paid to endorse it.